You're standing in a grocery store, holding two identical-looking ears of corn. One has a small label that says non-GMO project verified. The other has no label at all. You turn them over in your hands, feeling their weight, examining their kernels. They look the same. They probably taste the same. But something about that little label makes you pause, makes you wonder what invisible difference might exist between these two ears of corn and whether it matters at all. Welcome back to Midnight Deep Dive. Or if you're new here, welcome to our cozy corner of the internet, where we explore the fascinating stories behind everyday things we often take for granted. Tonight, we're diving into one of the most debated topics in modern food: genetically modified organisms, or GMOs. But this isn't a story about taking sides or telling you what to think. Instead, we're going to trace the surprisingly long history of humans modifying their food, understand how modern genetic engineering actually works, and explore why something so scientifically mundane became so culturally controversial. So settle into your most comfortable position, let your breathing slow and deepen, and join me as we explore the seeds of change that transformed our world. Imagine you're living 10,000 years ago, somewhere in the Fertile Crescent of the Middle East. You're part of a small group of humans who have spent the day gathering wild grains from the grasses that grow in this temperate region. The work is tedious and the reward is small. These wild wheat plants have tiny seeds that shatter and scatter at the slightest touch, making them difficult to harvest. Most of your time is spent crawling on hands and knees, picking up individual seeds from the ground. But then you notice something. Among all these plants with their shattering seed heads, there's one that seems different. Its seeds cling to the stalk just a bit longer, making them easier to collect. You gather seeds from this plant, and perhaps without even thinking about it, you save some to scatter near your camp the next season. You've just taken the first step toward agriculture, and you've done something else too. You've begun the process of modifying your food. This is where our story of genetically modified organisms truly begins. Even though those ancient farmers would have had no concept of genetics, no understanding of heredity, no knowledge that they were changing the fundamental nature of the plants they depended on. They were simply selecting seeds from plants that had useful traits. Seeds that stayed on the stalk, grains that were larger, plants that grew more vigorously. Over generations, this simple practice of saving seeds from the best plants transformed wild grasses into the wheat, barley, and rice that would become the foundation of human civilization. The transformation these ancient farmers accomplished is almost unbelievable when you see it with modern eyes. Wild teosinte, the ancestor of modern corn, produces a single stalk with perhaps a dozen seeds, each encased in a hard shell that would break your teeth if you tried to eat it. It looks nothing like corn. Most people wouldn't even recognize it as related. Yet over thousands of years, through nothing but careful seed selection, indigenous peoples in Mexico transformed this barely edible grass into maize with large, soft-kerneled cobs. The genetic changes required for this transformation are so extensive that modern corn cannot survive without human cultivation. It's completely dependent on us to remove its seeds from the cob and plant them. Bananas tell an even stranger story. The wild ancestors of the bananas we eat today were small, hard fruits filled with large, black seeds that made them barely palatable. Ancient farmers in Southeast Asia, perhaps 7,000 years ago, noticed occasional banana plants that produced fruits with fewer seeds or sweeter flesh. They propagated these plants through cuttings, and over millennia, they created the seedless, sweet banana we recognize today. Modern commercial bananas are so modified from their wild ancestors that they're sterile. They produce no viable seeds at all. Every banana plant is a clone, genetically identical to every other banana plant of the same variety, propagated endlessly through human intervention. Watermelons in 17th century paintings look alien to modern eyes. Small, with large seed-filled sections, and very little of the sweet red flesh we associate with watermelon today. Just a few hundred years of selective breeding transformed them into the massive, mostly flesh, mostly seedless fruits we eat today. Carrots were originally purple, white, or yellow. The orange carrot we know was developed in the Netherlands in the 17th century, possibly to honor the Dutch royal family. Even broccoli, cauliflower, cabbage, kale, and Brussels sprouts are all the same species, Brassica oleracea, modified through selection to emphasize different parts of the plant. This practice of selecting and saving seeds from desirable plants became more sophisticated over time. Ancient farmers learned to cross-pollinate plants, taking pollen from one individual and deliberately applying it to another to combine traits they valued. This wasn't genetic engineering in the modern sense, but it was definitely genetic modification. Changing the genetic makeup of crops through human intervention and selection. Medieval farmers and gardeners developed the art of grafting, cutting branches from one plant and attaching them to the rootstock of another. This technique, still used today, allows farmers to combine the best traits of different varieties. The roots might be from a hardy, disease-resistant plant, while the branches produce delicious fruit. Walk through an apple orchard today, and virtually every tree is grafted. The science of grafting was a mystery to medieval practitioners, but they perfected the technique through careful observation and experimentation. By the 18th and 19th centuries, plant breeding had become a sophisticated practice. Gregor Mendel's famous experiments with pea plants in the 1860s revealed the mathematical patterns of inheritance, though his work wasn't widely recognized until the early 20th century. Once Mendel's laws were rediscovered and combined with the understanding that DNA carried genetic information, plant breeding became increasingly intentional and scientific. Agricultural scientists developed hybrid crops by crossing two different parent lines to produce offspring with desirable traits, often higher yields or greater uniformity. Hybrid corn, developed in the 1930s, dramatically increased food production. Farmers couldn't save seeds from hybrid plants because the next generation wouldn't have the same traits, creating the first crops where farmers had to purchase new seeds each year, a practice that would later become controversial in the GMO debate. Throughout the 20th century, scientists used various techniques to speed up the process of creating new crop varieties. Radiation breeding involved exposing seeds to radiation to cause random mutations, then selecting the rare mutations that produced useful traits. Chemical mutagenesis used chemicals to cause genetic changes. These techniques created thousands of new crop varieties, many of which are still grown today and are considered conventional or even organic. The ruby red grapefruit, for instance, was created through radiation breeding, as were many varieties of wheat, barley, and rice. What's remarkable about this history is that humans have been modifying the genetics of their food for as long as agriculture has existed. Every domesticated crop is genetically different, often dramatically different from its wild ancestors. We created these changes through selection, breeding, hybridization, and even mutation, all forms of genetic modification. The wheat, corn, bananas, and tomatoes we eat today are all products of human intervention in plant genetics. They're artificial in the sense that they wouldn't exist without us, and many couldn't survive without our continued cultivation. When we talk about GMOs today, we're really talking about a new method of accomplishing something humans have done for 10,000 years, changing plants to better serve our needs. The question isn't whether we should modify our food. We've been doing that since the dawn of agriculture. The question is about the methods we use, the speed of change, the types of modifications we can now make, and who controls this powerful technology. To understand those questions, we need to understand what came next. The Green Revolution and the promise that science could feed the world. In the 1960s, a quiet American scientist named Norman Borlaug was working in Mexican wheat fields. and he was about to help save perhaps 1 billion lives. This might sound like hyperbole, but it's probably an understatement. Borlaug's work and the broader agricultural revolution he helped create transformed food production so dramatically that it prevented the mass famines many experts had predicted were inevitable. The world in the mid-20th century faced a genuine crisis. Human population was growing rapidly, particularly in developing nations. Agricultural production in many regions was stagnant, using traditional methods that had changed little in centuries. Experts predicted catastrophic famines that would kill hundreds of millions of people. The question wasn't whether mass starvation would occur, but how terrible it would be. Paul Ehrlich's 1968 book, The Population Bomb, captured this pessimism, predicting that nothing could prevent massive die-offs in the 1970s and 1980s. Norman Borlaug didn't accept this inevitability. Working with Mexican agricultural researchers, he developed new varieties of wheat using traditional crossbreeding techniques, but with unprecedented intensity and purpose. His wheat varieties were semi-dwarf, meaning they grew shorter and sturdier, putting more energy into grain production rather than growing tall stalks. These plants could handle heavy applications of fertilizer without toppling over under the weight of their own grain. They were also disease-resistant and could grow in a wider range of climates. The results were extraordinary. Mexican wheat production soared. Borlaug then took his wheat varieties to India and Pakistan, countries facing the most immediate famine threats. Despite political turmoil, wars, and immense logistical challenges, the new wheat varieties were planted across millions of acres. Yields doubled, then tripled. India, which had been importing millions of tons of wheat annually and facing starvation, became self-sufficient in wheat production. Pakistan experienced similar transformations. The predicted famines didn't materialize. This wasn't just about wheat. The Green Revolution, as it came to be called, involved new varieties of rice, corn, and other staple crops. It included improved irrigation, increased fertilizer use, better pest management, and mechanization. Scientists at international research centers bred crops specifically for tropical and subtropical climates, where most of the world's hungry people lived. These weren't genetically engineered crops in the modern sense. They were developed through accelerated traditional breeding, but the intensity and scientific rigor were unprecedented. The impact was profound and global. Between 1960 and 2000, world grain production more than doubled, while the land used for agriculture increased by only about 10%. Asia, Africa, and Latin America dramatically increased food production. Malnutrition rates fell. Child mortality declined. The Green Revolution is credited with saving somewhere between 250 million and 1 billion lives, depending on how you calculate prevented deaths from famine. Norman Borlaug received the Nobel Peace Prize in 1970, one of the few times the prize has been awarded for scientific agricultural work. The Nobel Committee recognized that preventing famine was fundamentally a contribution to peace, as hunger and resource scarcity are often precursors to conflict. Borlaug himself was modest about his achievements, always emphasizing that the Green Revolution was the work of thousands of scientists, farmers, and policymakers working together. But the Green Revolution also revealed the limits of traditional plant breeding. Developing new varieties through crossbreeding required years or decades of work. Breeders could only work with traits that existed within a species or closely related species. If you wanted to add a trait to wheat that didn't exist anywhere in wheat's wild relatives, you were simply out of luck. The process was slow, imprecise, and limited by the genetic diversity available within breeding populations. Furthermore, the Green Revolution's success came with costs that became more apparent over time. The new varieties required heavy inputs of water, fertilizer, and pesticides. Small farmers who couldn't afford these inputs often didn't benefit. The focus on high-yielding varieties meant that thousands of traditional crop varieties were abandoned, reducing agricultural biodiversity. Intensive irrigation depleted aquifers. Fertilizer runoff polluted waterways. The environmental impact of industrial agriculture became increasingly concerning. By the 1980s and 1990s, agricultural scientists faced new challenges. Climate change was making weather patterns less predictable. Pests and diseases were evolving resistance to pesticides. Water resources were becoming scarcer. The gains from the Green Revolution were plateauing as traditional breeding approached biological limits. New diseases threatened crucial crops: rice blast, wheat rust, corn viruses. Traditional breeding had accomplished extraordinary things, but it was increasingly clear that new tools would be needed to address the challenges ahead. During these same decades, molecular biology was experiencing its own revolution. In 1953, Watson and Crick discovered DNA's double helix structure, revealing how genetic information was stored. Throughout the 1960s and 70s, scientists learned to read DNA sequences, understanding the genetic code that determined how organisms developed and functioned. They discovered restriction enzymes that could cut DNA at specific sequences, and ligases that could paste DNA segments together. In the 1970s, scientists realized these tools could potentially do something extraordinary. They could move genes between organisms that would never naturally interbreed. They could take a gene from one species and insert it into another, giving the recipient organism entirely new capabilities. A bacterial gene could be added to a plant. A fish gene could be added to a tomato. The barriers that had limited traditional breeding, the requirement that organisms be closely related enough to interbreed, could potentially be overcome. This was the moment when genetic modification became something qualitatively different from the selective breeding humans had practiced for millennia. Instead of selecting from existing genetic variation or waiting for useful mutations to occur naturally, scientists could now intentionally design organisms with specific new traits. They could do in years or even months what traditional breeding might take decades or centuries to accomplish, if it could accomplish it at all. The promise was extraordinary. Crops could be engineered to resist devastating diseases, to tolerate drought or salty soils, to produce more nutritious food, to require fewer pesticides, to adapt to climate change. The Green Revolution had shown that improved crops could save lives and transform societies. Genetic engineering seemed to offer tools that could extend these benefits far beyond what traditional breeding could achieve. But this new power also raised profound questions. Was it safe to move genes between species that nature had kept separate? Would these modified organisms affect ecosystems in unpredictable ways? Who would control this technology, and would it serve the needs of hungry people or primarily corporate profits? Was there something fundamentally different, perhaps something wrong, about this kind of genetic modification compared to traditional breeding? These questions would define the debate over GMOs for decades to come. But before we explore that debate, we need to understand what genetic engineering actually is, how it works, and what it can and cannot do. The science itself is less scary and more fascinating than many people realize, and understanding it helps us think more clearly about both the promises and the concerns. So let's step into the laboratory and see how scientists actually create a genetically modified organism. There's something almost magical about the idea of moving genes between species. It sounds like science fiction, like playing God, like tampering with the fundamental fabric of life. But when you understand how genetic engineering actually works, it becomes less mystical and more like very sophisticated gardening. The process is complex and requires years of training, but the basic principles are surprisingly straightforward. And perhaps most importantly, nature itself has been doing something similar for billions of years. Let's start with what genes actually are. Your body and the bodies of all living things are made of cells. Inside most of those cells is a nucleus, and inside that nucleus are chromosomes, long strands of DNA twisted into their famous double helix shape. DNA is essentially a very long instruction manual written in a chemical alphabet with just 4 letters: A, T, G, and C. These letters, strung together in specific sequences, form genes. Instructions for making proteins, which are the machinery that actually does the work of living. Every living thing, from bacteria to bananas to humans, uses the same basic system. We all use DNA, we all use the same genetic code, we all produce proteins the same way. This universality is one reason genetic engineering is possible. Because all life uses the same fundamental language, a gene from one organism can be read and used by a completely different organism. It's like all living things are running the same basic operating system, even if they're running very different applications. Traditional plant breeding works by shuffling the genes that already exist within a species or between very closely related species. When 2 wheat plants are crossed, their offspring get a mix of genes from both parents. Breeders can select offspring with desirable combinations of genes, then breed those plants again, gradually accumulating beneficial traits. But this process has limitations. You can only work with genes that exist within the breeding population, and you can't control which genes get passed along together. It's like shuffling a deck of cards. You can get different combinations, but you can't add new cards to the deck. Genetic engineering, by contrast, allows scientists to add entirely new genes to the deck. Here's how it typically works. Let's say researchers want to create a corn plant that's resistant to a particular insect pest. They might start by identifying a gene from a soil bacterium called Bacillus thuringiensis, or BT for short. This bacterium produces a protein that's toxic to certain insects, but completely harmless to humans and most other organisms. Organic farmers have been spraying BT bacteria on their crops as a natural pesticide for decades. The researchers would first isolate the BT gene, the specific section of bacterial DNA that contains instructions for making the insect-toxic protein. Using restriction enzymes, they can cut this gene out of the bacterial DNA. Think of restriction enzymes as very precise molecular scissors that cut DNA only at specific sequences. They then need to insert this gene into corn DNA. There are several methods for getting new genes into plant cells. One early technique was literally called a gene gun. Scientists would coat tiny gold particles with copies of the desired gene, then use compressed gas to shoot these particles into plant cells. Some particles would penetrate cell nuclei, and the DNA would integrate into the plant's chromosomes. It sounds almost comically crude, and the success rate was low, but it worked often enough to be useful. A more elegant method uses a natural genetic engineer, Agrobacterium tumefaciens. This soil bacterium has evolved the ability to insert some of its own genes into plant cells, causing the plant to produce tumors that the bacteria can use for food. Scientists discovered they could hijack this natural gene transfer system, replacing the tumor-causing genes with whatever gene they wanted to insert. The bacterium becomes a delivery vehicle, infecting plant cells and inserting the desired gene into the plant's DNA. Once a gene is successfully inserted into a plant cell, researchers need to grow that single modified cell into a whole plant. This is where plant tissue culture comes in, a technique that's been used in conventional breeding for decades. Under the right conditions of nutrients, hormones, and light, a single plant cell can be coaxed to develop roots, then shoots, eventually becoming a complete plant. This regenerated plant will have the new gene in every cell, and it will pass that gene to its offspring. But adding a gene is just the beginning. The researchers need to verify that the gene actually works, that it's producing the desired protein, that the protein is having the intended effect, that it's not causing unexpected problems. They grow the modified plants under controlled conditions, testing them extensively. Does the plant really resist the target insect? Does it grow normally otherwise? Are there any unintended changes in the plant's chemistry? This testing process can take years. Most genetically engineered plants don't include just one gene. They typically include several, the main gene providing the desired trait, a marker gene that helps researchers identify which plants successfully incorporated the new DNA, and regulatory sequences that control where and when the new gene is active. You wouldn't want the Bt protein being produced in every cell all the time. That would waste the plant's energy. Instead, regulatory sequences might ensure the protein is only produced in the parts of the plant that insects eat. It's important to understand what genetic engineering can and cannot do. It's very good at adding specific, well-understood traits encoded by one or a few genes. Insect resistance, herbicide tolerance, disease resistance, vitamin production. These traits can be successfully engineered because they're controlled by a small number of genes with clear functions. But more complex traits like drought tolerance, yield increase, or improved flavor might involve dozens or hundreds of genes interacting in complicated ways. Genetic engineering can contribute to these traits, but it's not a magic wand that can redesign an organism from scratch. Modern genetic engineering has become more sophisticated with the development of CRISPR technology in the 2010s. CRISPR allows scientists to edit genes with unprecedented precision, not just adding new genes, but modifying existing ones, deleting unwanted sequences, or making tiny changes to specific DNA letters. Think of traditional genetic engineering as writing a new paragraph and inserting it somewhere in a book, while CRISPR is more like using find and replace to change specific words throughout the text. CRISPR has sparked new debates about what counts as a GMO. If you use CRISPR to make a change in a plant that could have occurred through natural mutation or traditional breeding, is the result a GMO? Different countries have answered this question differently. Some regulate CRISPR-edited plants as GMOs, while others don't, focusing instead on the final product rather than the process used to create it. Here's something that often surprises people. Nature has been doing horizontal gene transfer, moving genes between unrelated organisms for billions of years. Bacteria trade genes constantly, which is one reason antibiotic resistance can spread so quickly. Viruses incorporate their genetic material into host cells, and sometimes those viral genes become permanent parts of the host's DNA. About 8% of human DNA consists of ancient viral sequences. Some plants have acquired genes from bacteria through natural processes. The genetic engineering that scientists do in labs is unprecedented in its precision and intention, but gene transfer between species is a natural phenomenon, not a purely human invention. The science of genetic engineering is neither as magical nor as terrifying as it's sometimes portrayed. It's a sophisticated technique for doing something humans have done since the dawn of agriculture, modifying crops to better serve our needs, but with new precision and new possibilities. A genetically engineered plant is still a plant. It grows in soil, uses sunlight for photosynthesis, produces seeds or fruits. It's not a Frankenstein monster or an alien organism. It's a plant with one or a few new genes, created through human ingenuity rather than human patience. Understanding this science doesn't tell us whether genetic engineering is good or bad, whether it should be embraced or restricted. Those are separate questions involving values, priorities, and how we weigh benefits against risks. But understanding the science helps us think clearly about those questions, separating genuine concerns from science fiction fears. And it prepares us to understand what happened when these scientifically engineered crops left the laboratory and entered the food supply, which is where our story goes next. In 1994, a tomato called the flavor saver arrived in American grocery stores, and it represented something unprecedented in the 10,000-year history of agriculture. This wasn't a tomato bred through generations of careful selection. It wasn't a hybrid or a mutant variety. It was the first commercially available genetically engineered whole food created by a company called Calgene, and it was designed to solve one of the most frustrating problems in modern produce: the mushy, flavorless tomato. The problem Calgene was trying to address was simple but significant. Tomatoes are picked green and hard so they can survive shipping without turning to mush. They're then artificially ripened with ethylene gas, producing the pale, mealy tomatoes most of us know from grocery stores. Vine-ripened tomatoes are delicious, but they're too soft to ship long distances. Calgene's solution was elegant. They identified the gene responsible for producing an enzyme that softens tomatoes as they ripen, and they essentially turned down its volume. The flavor saver tomato could ripen on the vine, developing full flavor, but it would stay firm enough to ship. The flavor saver made it through extensive testing by the FDA and USDA. Calgene voluntarily labeled it as genetically engineered, betting that consumers would embrace the improved taste. The company invested enormous resources in bringing this tomato to market. Over $20 million in development costs, years of regulatory navigation, and millions more in marketing. The tomatoes were priced at a premium, positioned as a superior product worth the extra cost. The flavor saver failed commercially. The reasons were complicated and had little to do with genetic engineering itself. The tomatoes didn't ship as well as hoped, they were expensive to produce, and Calgene lacked the agricultural expertise to grow them successfully at scale. The company was eventually bought by Monsanto, and the flavor saver quietly disappeared from stores by 1997. But its legacy was significant. It proved that genetically engineered food could be approved, produced, and sold. The door was open for what came next. What came next was dramatically different in both approach and impact. Instead of focusing on food quality improvements that consumers would directly experience, the next wave of GMO crops offered benefits to farmers, traits that made crops easier or cheaper to grow. And instead of small biotech startups like Calgene, the companies developing these crops were agricultural giants with the resources to bring products to market at massive scale. In 1996, Monsanto introduced Roundup Ready soybeans. These beans were engineered to tolerate glyphosate, the active ingredient in Monsanto's Roundup herbicide. The innovation was simple but revolutionary for farmers. Traditionally, controlling weeds without harming crops required either extensive manual labor, expensive mechanical cultivation, or carefully timed applications of multiple herbicides that would kill weeds but not crops. Roundup Ready soybeans simplified everything. Farmers could plant their crops, then spray Roundup directly over the entire field. The weeds died, the soybeans thrived. For farmers, especially those managing large acreage, this was transformative. Weed control became easier and often cheaper. Farmers could use no-till farming practices, leaving crop residue from previous seasons on the field instead of plowing, which reduced soil erosion and fuel costs. Monsanto positioned Roundup Ready crops as environmentally friendly because they supposedly allowed farmers to use less total herbicide and reduce the environmental impact of tillage. Adoption was extraordinarily rapid. By 2014, over 90% of soybeans grown in the United States were Roundup Ready. The trait was soon engineered into corn, cotton, canola, and other crops. Farmers worldwide embraced the technology because it solved real problems they faced. The market had spoken, and it said that farmers valued this innovation. Monsanto also introduced Bt crops, starting with corn in 1996. These crops contained genes from Bacillus thuringiensis, the soil bacterium that produces proteins toxic to certain insects. Bt corn produced these insecticidal proteins in its own tissues, meaning that when corn borers or other target insects ate the plant, they died. Farmers could reduce their use of synthetic insecticides, avoiding the costs, labor, and environmental impact of spraying. The benefits seemed clear and substantial. Studies showed that Bt crops reduced insecticide use by millions of pounds annually in the United States alone. Farmers saved money on pesticides and saw reduced crop losses to insect damage. In developing countries like India and China, where small farmers often couldn't afford expensive insecticides, Bt cotton promised to protect crops from devastating pests while reducing farmer exposure to toxic chemicals. But these first-generation GMO crops also established patterns that would become controversial. The crops didn't directly benefit consumers. They benefited farmers and seed companies. Consumers ate Roundup Ready soybeans and Bt corn, mostly in processed form, without noticing any difference in taste, quality, or price. The value proposition was entirely on the production side. Moreover, the crops were typically paired with proprietary agricultural chemicals from the same companies that sold the seeds. Monsanto sold both Roundup Ready seeds and Roundup herbicide. This vertical integration raised questions about market control and farmer independence. The seeds themselves were patented, and farmers who purchased them signed agreements not to save seeds for replanting, a practice farmers had followed for thousands of years. They had to purchase new seeds each season, creating a recurring revenue stream for seed companies. These business practices weren't entirely new. Hybrid corn had already established that farmers would purchase seeds annually rather than saving them, and patents on new plant varieties had existed for decades through the Plant Variety Protection Act. But the combination of genetic engineering, patents, and contractual restrictions on seed saving created unease about corporate control over food production. When Monsanto sued farmers for patent infringement after finding patented genes in their fields, sometimes in plants that may have grown from seeds that had blown from neighboring fields, the legal actions generated significant public backlash. Nevertheless, adoption continued to accelerate globally. By the early 2000s, GMO crops were being grown on hundreds of millions of acres across the Americas, Asia, and other regions. The crops that dominated were soybeans, corn, cotton, and canola, almost all featuring either herbicide tolerance or insect resistance, or both traits stacked together in the same plants. These four crops represented over 99% of GMO acreage worldwide. Other genetically engineered crops were developed but never commercialized, or were approved but never widely adopted. Virus-resistant papaya saved Hawaii's papaya industry from a devastating disease in the 1990s and remains a GMO success story. But it's a niche crop representing a tiny fraction of GMO acreage. Bt eggplant was developed and approved in Bangladesh to help small farmers combat a destructive pest. But adoption has been limited by regulatory challenges and public skepticism. The pattern became clear. Genetically engineered crops succeeded commercially when they offered clear economic value to farmers, when they could be grown at massive scale, and when they integrated into existing industrial agriculture systems. Crops that might have benefited consumers directly. The better-tasting tomato, more nutritious fruits, longer-lasting vegetables, largely didn't make it to market, partly because of commercial failures like the Flavor Saver, but also because of the increasing regulatory costs and public skepticism that made consumer-focused GMOs harder to commercialize. By 2000, most processed foods in American grocery stores contained ingredients from GMO crops, primarily corn and soy derivatives. High-fructose corn syrup, soybean oil, cornstarch, soy lecithin. These ingredients were ubiquitous in processed foods, and most came from GMO crops. Yet few consumers knew they were eating GMO-derived foods because labeling wasn't required. The foods looked the same, tasted the same, and cost the same as conventional alternatives. This invisibility to consumers, combined with the concentration of benefits among farmers and seed companies, would contribute to the public skepticism that was beginning to build. If consumers weren't experiencing obvious benefits, if the technology was mainly making life easier for industrial agriculture rather than feeding hungry people, if control was concentrated in the hands of a few large corporations, then why should the public embrace genetic engineering? This question would become more pressing as stories of humanitarian applications for GMO technology emerged. Particularly one crop that promised to prevent childhood blindness but would become mired in controversy for over 2 decades. In the mid-1990s, 2 scientists, Ingo Patricus and Peter Bayer, were working on what they hoped would be a transformative humanitarian project. They wanted to engineer rice to produce beta-carotene, the orange pigment found in carrots that the human body converts into vitamin A. Their goal wasn't profit or agricultural efficiency. They wanted to prevent blindness and death in children. The problem they were addressing is genuinely horrifying in its scope. Vitamin A deficiency is one of the world's most serious nutritional problems, affecting millions of people, mostly in Asia and Africa, where rice is the staple food. The consequences are devastating. Between 250,000 and 500,000 children go blind every year due to vitamin A deficiency, and about half of those children die within a year of losing their sight. The deficiency also weakens immune systems, making children more susceptible to infectious diseases like measles and diarrhea that might otherwise be survivable. The cruel irony is that rice plants produce beta-carotene in their leaves and other green parts, where it's needed for photosynthesis. But the genes that produce beta-carotene are switched off in the rice grain, the part people eat. White rice, which has had its outer layers removed during milling, contains essentially no vitamin A or its precursors. People who depend on rice for most of their calories, particularly children and pregnant women, often become vitamin A deficient. The obvious solution would be dietary diversification, adding foods rich in vitamin A like eggs, dairy, vegetables, and fruits. But for the world's poorest people, particularly those in rural areas with limited access to diverse foods, this isn't always feasible. Supplementation programs, where children receive high-dose vitamin A capsules once or twice a year, have helped significantly. But they require extensive infrastructure, healthcare access, and continued funding. They also don't reach everyone who needs them. Patricus and Bayer envisioned a different approach. What if rice itself could provide vitamin A? What if the genes for beta-carotene production in rice grains could be switched back on, or new genes added to create the biosynthetic pathway? People could grow it, eat it, and get the vitamin A they needed from their staple food. No behavior change required beyond choosing these seeds. The technical challenge was significant. Producing beta-carotene in rice grains required not just one gene but several, working together in a biosynthetic pathway. The scientists needed to introduce genes from daffodils and bacteria, along with regulatory sequences to ensure they functioned properly. After years of work, they succeeded in 1999, creating rice with grains that were distinctly golden orange from the beta-carotene they contained. They called it golden rice. The scientists made an extraordinary decision. They would make golden rice available to subsistence farmers for free. They worked with seed companies and negotiated complex patent licenses for all the technologies involved, but their goal was humanitarian, not commercial. Farmers earning less than $10,000 per year would be able to save and replant seeds without restrictions. Golden rice would be given away, not sold. The scientific community largely celebrated this achievement. Time magazine featured golden rice on its cover in 2000, calling it a potential solution to malnutrition affecting millions. Agricultural scientists saw it as exactly the kind of beneficial application that genetic engineering should pursue, addressing genuine human suffering rather than just corporate profits. Here was proof that GMO technology could serve humanitarian goals. But golden rice would not reach farmers quickly. In fact, over 2 decades after its creation, it's still not widely available. The story of why reveals the complex interplay of regulation, politics, activism, and genuine scientific concerns that have defined the GMO debate. First, there were technical improvements to make. The original golden rice didn't produce as much beta-carotene as researchers hoped. A second version, Golden Rice 2, developed in 2005, produced 23 times more beta-carotene than the original. Studies had to confirm that eating golden rice actually increased vitamin A levels in humans. Field trials needed to verify that it grew well in various environments and that the beta-carotene remained stable during storage and cooking. Then came the regulatory requirements. Each country where golden rice might be grown needed to evaluate its safety and approve it for cultivation and consumption. These regulatory processes, designed to ensure GMO safety, could take years and cost millions of dollars. Resources that humanitarian projects struggled to secure. The Philippines, where golden rice research has been most advanced, began the approval process in 2008. But approval for direct use as food didn't come until 2021. Approval for cultivation took even longer. But the most significant obstacles to golden rice weren't technical or regulatory. They were political and ideological. Environmental organizations, particularly Greenpeace, opposed golden rice from the beginning. Their arguments were multifaceted. Some contended that golden rice was a distraction from more effective solutions like dietary diversification and supplementation programs. Others argued it would benefit primarily seed companies and biotech corporations, not poor farmers. Some claimed it was a public relations tool designed to improve the image of genetic engineering and make people more accepting of GMO crops generally. Greenpeace and other organizations argued that golden rice was unnecessary because other vitamin A interventions were working. They pointed to supplementation programs that had reduced vitamin A deficiency rates. Why rush a genetically engineered solution when conventional approaches were succeeding? The activists also raised concerns about safety, environmental impacts, and the precedent of introducing GMO crops for humanitarian reasons when commercial crops remained controversial. These arguments found resonance, particularly in countries where GMO crops were already contentious. In some regions, activists destroyed field trials of golden rice, literally uprooting test plots to prevent research from continuing. A 2013 incident in the Philippines, where activists destroyed a golden rice field trial, made international news and polarized opinions further. Supporters saw it as vandalism that set back humanitarian research. Opponents saw it as justified resistance to corporate agricultural technology. Scientists who supported golden rice became increasingly frustrated. In 2016, 110 Nobel laureates signed an open letter supporting golden rice and criticizing Greenpeace's opposition. The letter accused opponents of denying crucial nutrition to the world's poor based on emotion and ideology rather than evidence. It called for an end to opposition to GMO crops in general and golden rice in particular. The response from GMO critics was equally impassioned. They argued that scientists were oversimplifying a complex issue, ignoring legitimate concerns about agricultural systems and corporate control, and presenting golden rice as a silver bullet when more comprehensive approaches to malnutrition were needed. The debate revealed deep divides not just about golden rice specifically, but about technology, development, corporate power, and how to address global poverty. The tragedy is that while these arguments continued, children kept going blind and dying from vitamin A deficiency. An estimated 3 to 4 million children have died from vitamin A deficiency in the time since golden rice was first developed. Whether golden rice would have saved those lives is debatable. Implementation would have taken time, and supplementation programs were already helping many children. But the possibility that a beneficial technology was delayed by a combination of overregulation and activist opposition weighs heavily on many scientists. As of the early 2020s, golden rice is finally being approved in some countries and may soon be planted by farmers. The Philippines approved it for cultivation in 2021. Bangladesh is also moving toward approval. Whether it will significantly reduce vitamin A deficiency remains to be seen. Supplementation programs have improved, dietary habits have changed, and the problem, while still serious, isn't as severe as it was in 2000. The golden rice story reveals uncomfortable truths about GMO technology. It shows that even when genetic engineering is applied to humanitarian goals, with no profit motive, with free access for poor farmers, the technology remains controversial. It demonstrates how regulatory caution, even when well-intentioned, can delay beneficial innovations. It highlights how activist opposition, even when rooted in genuine concerns about corporate agriculture, can prevent potentially life-saving technologies from reaching those who need them. But it also shows why simple narratives about GMOs being purely good or purely bad don't capture reality. Questions about whether golden rice is the best solution to vitamin A deficiency are legitimate. Concerns about dependency on single technological solutions rather than building diverse, resilient food systems are worth considering. The debate reveals that discussions about GMO technology can't be separated from broader questions about development, agriculture, corporate power, and how we address global inequality. Golden rice promised to be a clear-cut humanitarian success story for genetic engineering. Instead, it became another battleground in the larger GMO war, with children's lives caught in the crossfire. The complexity of this case prepares us to understand the broader debate about GMOs that has raged for decades. A debate that involves science, but extends far beyond it into questions of trust, values, and competing visions of how agriculture should be organized. Imagine two intelligent, well-informed people sitting down to discuss GMO foods. One is a plant geneticist who has spent decades researching crop improvement. The other is an organic farmer and environmental activist. If you listen to their conversation, you might start to wonder if they're discussing the same topic at all. They cite different evidence, emphasize different concerns, and reach completely opposite conclusions about whether GMO crops should be embraced, tolerated, or banned. This isn't because one is smart and the other foolish, or because one is honest and the other lying. It's because the GMO debate has never been purely about science. The scientific controversy about GMOs, to the extent one exists within the research community, is relatively narrow. Thousands of studies over 3 decades have examined whether GMO foods are safe to eat, whether GMO crops harm the environment, and whether genetic engineering creates unexpected effects in organisms. The overwhelming consensus among scientists, scientific organizations, and regulatory agencies is that currently approved GMO crops are safe to eat and are no riskier than conventional crops. The American Medical Association, the American Association for the Advancement of Science, the World Health Organization, the European Commission, the National Academy of Sciences, and dozens of other prestigious scientific bodies have all concluded that GMO foods currently on the market are safe for human consumption. These aren't industry-funded groups. They're independent scientific organizations evaluating the research without financial interest in the outcome. Studies have fed GMO foods to animals across multiple generations, looking for any health effects. They've examined whether proteins produced by GMO crops cause allergic reactions. They've tested whether the genetic modification process creates unexpected toxins or allergens. They've compared the nutritional content of GMO and non-GMO crops. The conclusion, repeated across hundreds of studies and systematic reviews, is that eating currently approved GMO crops is no more dangerous than eating conventional crops. This scientific consensus is about as strong as the consensus on climate change or the safety of vaccines. That doesn't mean every single scientist agrees. You can always find individual researchers with dissenting views. And it doesn't mean genetic engineering is risk-free or that every possible GMO would be safe. It means that the GMO crops that have been approved and commercialized after extensive testing don't pose unique risks to human health compared to conventional foods. So why, if the scientific consensus is so clear, does public skepticism about GMOs remain so widespread? Why do surveys consistently show that a majority of Americans believe GMO foods are unsafe, despite what scientific organizations say? Why have several European countries banned GMO cultivation? And why do many food companies advertise their products as non-GMO as a selling point? The answer is that the GMO debate has never been primarily about safety data or toxicology studies. It's been about trust, values, corporate power, and fundamentally different visions of how agriculture should be organized and who should control food production. Many environmentalists opposed GMOs from the beginning, not primarily because they thought the foods were poisonous, but because genetic engineering represented industrialized, corporate-controlled, reductionist agriculture, everything they opposed philosophically. GMO crops were developed by large corporations like Monsanto, patented and sold for profit, designed to work with industrial farming systems that relied on monocultures and chemical inputs. For people who believed agriculture should be small-scale, organic, diverse, and farmer-controlled, GMOs symbolized the wrong path regardless of whether they were technically safe to eat. These concerns weren't irrational. Monsanto's business practices, including suing farmers for patent infringement and aggressively protecting its market position, generated significant hostility. The consolidation of the seed industry, with a few large corporations controlling an increasing share of global seed markets, raised legitimate worries about corporate power over food systems. The fact that Roundup Ready crops encouraged heavy use of Roundup herbicide, benefiting the company that sold both products, looked suspiciously like a self-serving business model rather than a humanitarian innovation. Environmental organizations like Greenpeace, Friends of the Earth, and others mounted sophisticated campaigns against GMOs throughout the 1990s and 2000s. Their messaging often emphasized safety concerns and environmental risks, but the deeper opposition was ideological. They saw genetic engineering as a dangerous path that would increase corporate control, reduce biodiversity, create dependencies among farmers, and distract from more sustainable agricultural approaches. Some of this opposition drew on the precautionary principle, the idea that when an innovation has uncertain consequences, the safe approach is to avoid it until proven harmless. European policymaking has generally embraced this principle more than American regulation, which tends to allow innovations unless they're proven harmful. The different regulatory philosophies help explain why GMO cultivation is widespread in the Americas but rare in Europe. The precautionary principle sounds reasonable, but it has critics who point out that avoiding new technologies also has risks. If GMO crops could reduce pesticide use, increase food production, or help farmers adapt to climate change, then preventing their use could cause harm. The question becomes, what are we being cautious about, and what risks are we accepting by being cautious? There's no risk-free option, only different risk profiles. Consumer advocates added their voices to the debate, focusing on labeling. Many argued that regardless of safety, consumers had the right to know whether their food contained GMO ingredients. The resistance from food companies and agricultural corporations to mandatory labeling seemed suspicious. If GMOs were safe and beneficial, why oppose letting consumers know they were present? The labeling debate became a proxy for broader questions about transparency, consumer rights, and corporate accountability. The food industry's response to consumer concerns was pragmatic. Major brands started reformulating products to avoid GMO ingredients, not because they believed GMOs were unsafe, but because non-GMO had become a marketable claim worth the extra cost. Whole Foods and other retailers positioned themselves as GMO-free zones, attracting customers who distrusted industrial agriculture. The Non-GMO Project, which verifies and labels products as free from genetic engineering, became surprisingly successful, with thousands of certified products despite the organization having no regulatory authority. Meanwhile, farmers who grew GMO crops largely didn't engage in these philosophical debates. They made decisions based on what worked for their operations. If Roundup Ready soybeans made weed control easier and cheaper, they planted them. If Bt corn reduced insect damage and eliminated insecticide spraying, they used it. The technology adoption rates among farmers tell a different story than public opinion polls. Farmers voted with their planting decisions, and they voted overwhelmingly for GMO crops where they were available. But farmers in some regions also began experiencing problems that GMO critics had warned about. Superweeds evolved. Weed species that developed resistance to glyphosate through natural selection, just as bacteria develop antibiotic resistance. These weeds could no longer be controlled with Roundup alone, forcing farmers to use additional herbicides or return to mechanical weed control. Insect pests developed resistance to Bt toxins in some areas, reducing the effectiveness of Bt crops. These weren't unpredicted outcomes. Scientists knew resistance would eventually develop, but they happened faster in some regions than expected. These resistance problems didn't make GMO crops useless, but they complicated the narrative that genetic engineering would permanently solve agricultural problems. They suggested that GMO technology was a tool that could be valuable, but that needed to be used carefully as part of integrated pest management strategies, not as a silver bullet that eliminated the need for other approaches. The debate also became entangled with misinformation on both sides. Some GMO opponents circulated studies claiming to show health harms from GMO foods, but these studies were often methodologically flawed, based on small sample sizes, or failed to be replicated by other researchers. A notorious 2012 study by Gilzeric Seralini claimed that rats fed Roundup Ready corn developed tumors. But the study used a rat strain prone to tumors, had too few animals per group to draw statistical conclusions, and was eventually retracted for methodological flaws before being republished in a different journal. Similarly, GMO proponents sometimes oversold the benefits of genetic engineering, making promises that weren't fully realized. Claims that GMO crops would dramatically reduce world hunger, that they would substantially reduce pesticide use globally, or that they would be environmentally friendly miracle solutions often didn't match the complex reality. The first generation of GMO crops provided real but limited benefits, mostly to farmers in industrial agriculture systems, not revolutionary solutions to global food challenges. Social media and the internet amplified both good information and misinformation, making it increasingly difficult for people without scientific training to assess claims. Memes circulated showing horrifying images of supposed GMO, mutant, foods that were either photoshopped, ornamental varieties, or results of plant diseases. None actually GMOs. Simultaneously, social media allowed coordinated campaigns against GMOs to reach millions of people with emotionally compelling but scientifically questionable content. The tragedy of the GMO debate is that it became so polarized that nuanced positions became difficult to maintain. You were either pro-GMO or anti-GMO, either with science or against it, either supporting corporate agriculture or defending sustainable food systems. The middle ground, acknowledging that genetic engineering is a tool that can be used well or poorly, that current GMO crops have both benefits and drawbacks, that corporate control is a legitimate concern separate from safety issues. This nuanced position got lost in the shouting. What the debate revealed was a broader crisis of trust. Trust in scientific institutions, in regulatory agencies, in corporations, in government. People who distrusted these institutions were unlikely to believe their reassurances about GMO safety, regardless of how many studies supported that conclusion. People who felt that their concerns about corporate agriculture were dismissed as unscientific became more skeptical of scientific authority generally. The GMO debate became a lens through which deeper conflicts about power, expertise, and social values played out. Understanding this debate prepares us to examine one of the most personal questions about GMOs. Are they actually safe to eat? What does the science really tell us? And why do smart people sometimes reach different conclusions even when looking at the same data? Let's imagine you're standing in a grocery store holding 2 cans of corn. One is labeled non-GMO, the other isn't labeled at all, which probably means it contains GMO corn. You want to make a healthy choice for your family. You've heard people say GMOs are dangerous, and you've heard others say they're perfectly safe. What does the science actually tell us? The starting point for understanding GMO safety is recognizing what genetic modification does and doesn't do. When scientists insert a gene into corn to make it produce Bt protein, they're adding a new protein to the plant. Proteins are chains of amino acids, the same building blocks that make up proteins in every food you eat. Your body breaks down proteins into amino acids during digestion, whether those proteins came from a GMO plant, a conventional plant, or an animal. The process doesn't change based on how the protein was originally created. Safety testing for GMO crops follows a comparative approach. Regulators don't test whether GMO corn is safe in some absolute sense. That would be an impossible standard, as nothing is absolutely risk-free. Instead, they ask, is this GMO corn substantially equivalent to conventional corn? Does it have the same nutritional profile? Does it contain any new allergens or toxins? Are there any unexpected chemical changes resulting from the genetic modification? The testing process is extensive. Before a GMO crop can be commercialized, it undergoes years of evaluation. Scientists analyze its chemical composition, measuring proteins, fats, carbohydrates, vitamins, minerals, and thousands of other compounds. They compare these measurements to conventional varieties. If something unusual appears, it gets investigated. They test whether the new proteins cause allergic reactions using established allergenicity screening protocols. They feed the GMO crops to animals, looking for any adverse health effects across multiple generations. This testing isn't just done by the companies developing GMO crops. Regulatory agencies in multiple countries conduct independent evaluations. In the United States, the FDA, USDA, and EPA all review different aspects of GMO crops. In Europe, the European Food Safety Authority conducts additional assessments. These agencies don't simply accept company data. They evaluate the methodology, look at the raw data, and sometimes require additional studies before approval. After over 25 years of people eating GMO foods, and with billions of meals containing GMO ingredients consumed worldwide, we have a substantial body of evidence about safety. Long-term epidemiological studies have looked for health effects in populations that consume GMO foods compared to those that don't. These studies haven't found health differences attributable to GMO consumption. Animal feeding studies have been particularly extensive. Researchers have fed GMO crops to cattle, pigs, chickens, fish, rats, mice, and other animals, often across multiple generations, looking for any health effects. A comprehensive review published in 2014 examined 29 years of livestock feeding studies involving billions of animals. The conclusion was clear. Animals fed GMO crops showed no health differences compared to animals fed conventional crops. Critics sometimes point to individual studies claiming to show health effects from GMO foods. The Seralini study mentioned earlier is the most famous example. It reported that rats fed Roundup Ready corn developed tumors at higher rates than control rats. When the study was published in 2012, it received enormous media attention and seemed to vindicate concerns about GMO safety. But the scientific community quickly identified serious problems with the study. The rat strain used, Sprague Dawley rats, spontaneously develops tumors at high rates as they age. The study used only 10 rats per group, too few for statistical reliability with such a tumor-prone strain. The control group, rats, also developed tumors, but the study's presentation emphasized GMO-fed rats. The statistical analysis was questionable, with multiple comparisons made without correction, virtually guaranteeing that some would show significant differences by chance alone. The journal that published the study eventually retracted it, an unusual step that reflected the severity of the methodological flaws. Seralini republished it in a different journal, but numerous scientific organizations and regulatory agencies concluded that the study didn't provide credible evidence of GMO health risks. Multiple attempts to replicate the findings failed to show the health effects Seralini reported. This case illustrates an important principle. Not all studies are created equal. Individual studies can have flaws, use inappropriate methods, or reach conclusions that don't hold up under scrutiny. What matters is the weight of evidence. What does the broader body of research, particularly well-designed studies and systematic reviews, tell us? Systematic reviews and meta-analyses, which combine results from multiple studies to look for overall patterns, have consistently found no evidence that GMO foods cause health problems. These reviews have examined hundreds of studies, covering billions of meals and multiple animal species. The consensus conclusion is that GMO crops currently on the market are as safe to eat as their conventional counterparts. But what about allergenicity? Could GMO crops introduce new allergens? This is taken seriously in GMO testing. Proteins introduced through genetic engineering are screened against databases of known allergens. If a new protein resembles a known allergen, additional testing is required. The testing includes examining whether the protein is quickly broken down during digestion, whether it's stable when heated, and whether people with existing allergies react to it. One proposed GMO crop was actually prevented from reaching the market specifically because of allergenicity concerns. In the 1990s, a company developed soybeans with a gene from Brazil nuts to improve their protein quality for animal feed. Testing revealed that the new protein was allergenic to people with Brazil nut allergies. The product was immediately discontinued, never commercialized, and the soybeans were destroyed. This case is often cited as evidence that the safety testing system works. A potential problem was identified and prevented from reaching consumers. The question of long-term effects is often raised. How do we know that eating GMO foods for decades won't cause problems? This concern is understandable, but it applies equally to any food. Many conventional crops have been modified through radiation breeding or chemical mutagenesis, causing far more genetic changes than precise genetic engineering. Yet these crops aren't subjected to similar concerns about long-term effects. The ruby red grapefruit, created through radiation breeding, has been eaten for over 70 years without known health issues. Some critics argue that the genetic modification process itself might create unexpected changes in plants beyond the intended modification. This is a legitimate question that researchers have examined. Studies using comprehensive analysis techniques can compare thousands of compounds in GMO and conventional crops, looking for unexpected differences. These omics studies, genomics, proteomics, metabolomics, have found that GMO and conventional varieties are remarkably similar, with variation between different conventional varieties often being greater than variation between GMO and non-GMO versions of the same variety. Environmental growing conditions often have larger effects on plant chemistry than genetic modification. The same corn variety grown in different locations or different years will have more chemical variation than GMO versus non-GMO versions grown under identical conditions. This doesn't mean genetic modification causes zero unintended changes, but it suggests that any such changes are within the normal range of variation we accept in conventional crops. A common concern is whether eating GMO foods could somehow affect human genes. The logic is that if we're eating food with modified genes, could those genes get into our DNA? The answer is no, for several reasons. First, we eat DNA from every food. Conventional crops and animals are full of DNA. That DNA is broken down during digestion into individual nucleotides, the building blocks of DNA, which are chemically identical regardless of their source. Second, DNA from food doesn't integrate into human cells. If eating DNA changed our genes, we'd be a chaotic mix of traits from everything we've eaten. The question of whether glyphosate Roundup residues on Roundup Ready crops pose health risks is separate from GMO safety per se. This is about pesticide safety, which applies to both GMO and conventional crops. Glyphosate is one of the most extensively studied herbicides, with regulatory agencies worldwide having evaluated its safety. Most agencies have concluded it's safe when used according to label directions, though some studies have raised concerns about potential cancer risk at high exposure levels. The International Agency for Research on Cancer classified glyphosate as probably carcinogenic, while the EPA and European agencies concluded it's unlikely to be carcinogenic at typical exposure levels. This disagreement among agencies illustrates how scientific uncertainty gets interpreted differently depending on regulatory philosophy and what level of risk is considered acceptable. It's a separate debate from GMO safety, but it's often conflated because Roundup Ready crops are designed to be sprayed with glyphosate. Why do some people remain convinced that GMO foods are dangerous despite the scientific consensus? Several factors contribute. Naturalistic fallacy. The belief that natural is inherently better and safer than artificial makes genetic engineering seem wrong regardless of evidence. Motivated reasoning means people interpret new information to confirm existing beliefs. Trust in institutions matters. If you distrust regulatory agencies or believe they're captured by corporate interests, you won't believe their safety assessments. The GMO industry and its supporters haven't always helped their case. Dismissing concerns as unscientific or irrational alienates people with legitimate questions. Opposing labeling, even when safety isn't at issue, makes companies seem like they're hiding something. The concentration of GMO technology in a few large corporations, and those companies' often aggressive business practices, creates guilt by association. People transfer their distrust of Monsanto to the technology itself. The scientific consensus on GMO safety is about as clear as scientific consensus gets. That doesn't mean genetic engineering is risk-free or that every possible GMO would be safe. It means that the GMO crops currently on the market, after extensive testing and decades of consumption, haven't been shown to pose health risks different from conventional crops. For someone standing in that grocery store, deciding between GMO and non-GMO corn, the health considerations based on current evidence are minimal. Other factors: environmental impact, agricultural practices, corporate control, personal values about food systems might reasonably influence their choice, but health risk probably shouldn't be the deciding factor. Understanding what science tells us about safety helps us think more clearly about GMOs, but it doesn't resolve all the questions these technologies raise. The environmental impacts, the social and economic implications, and the future trajectory of genetic engineering still need examination. Our journey continues into the complexities of how GMO crops affect the world beyond our plates. There's a paradox at the heart of GMO crops' environmental story. When Bt corn was introduced, proponents celebrated that it would allow farmers to spray fewer insecticides. When Roundup Ready soybeans arrived, advocates pointed out that glyphosate was less toxic and less persistent than many alternative herbicides. The promise was that genetic engineering would make agriculture more environmentally friendly, and in some ways it has. But the reality, as with most agricultural technologies, turned out to be far more complicated than early promises suggested. Let's start with what has worked. Bt crops genuinely have reduced insecticide use in many contexts. Before Bt cotton was introduced in India and China, farmers often sprayed synthetic insecticides multiple times per season to control bollworms and other lepidopteran pests. These spraying operations were expensive, labor-intensive, and dangerous. Farmers, particularly small-scale farmers without proper protective equipment, experienced pesticide poisoning at alarming rates. Studies documented farmers getting sick, experiencing chronic health problems, and in some cases dying from pesticide exposure. Bt cotton changed this dramatically in many regions. The plants produced their own insecticidal proteins, killing target pests when they fed on the plants. Farmers who adopted Bt cotton reduced their insecticide applications significantly. Some studies showed reductions of 50% or more in synthetic insecticide use. This meant less chemical exposure for farmers, less environmental contamination, and lower costs. The environmental and health benefits were real and substantial. Similar patterns emerged with Bt corn in the United States and other countries. Farmers could reduce or eliminate insecticide spraying targeted at corn borers and other lepidopteran pests. This didn't just benefit farmers, it benefited the broader ecosystem. When farmers spray broad-spectrum insecticides, they kill not just pest insects, but also beneficial insects including pollinators, natural predators of pests, and insects that are simply part of the ecosystem. Reducing insecticide use meant less collateral damage to beneficial insect populations. But the story gets more complicated when we look at herbicide use with Roundup Ready crops. Initially, glyphosate use did replace more toxic herbicides, which was environmentally beneficial. Glyphosate breaks down relatively quickly in soil, doesn't accumulate in animal tissues, and is less toxic to many organisms than alternative herbicides like atrazine. The initial environmental impact seemed positive. However, the ease and effectiveness of using glyphosate led to increasing reliance on it. Farmers could spray their entire fields without worrying about damaging crops, and many did so repeatedly throughout the growing season. Total herbicide use in the United States increased substantially after Roundup Ready crops were introduced. By the 2010s, farmers were applying far more glyphosate than they had applied of all herbicides combined before Roundup Ready crops became widespread. This heavy reliance on a single herbicide created predictable evolutionary pressure. Weeds that happened to have genetic variations conferring even slight resistance to glyphosate survived, reproduced, and passed those traits to offspring. Within a decade of Roundup Ready crops being introduced, farmers began encountering glyphosate-resistant superweeds. Palmer amaranth, water hemp, and other species evolved resistance, spreading across millions of acres. These superweeds didn't make Roundup Ready crops useless, but they forced farmers to adapt. Some returned to tillage, which increased soil erosion and fuel use. Others began using additional herbicides, including older, more toxic ones that glyphosate was supposed to replace. The seed industry responded by developing crops with tolerance to multiple herbicides, stacking glyphosate resistance with resistance to dicamba or 24D. But this created new problems. Dicamba is prone to drift, damaging neighboring crops and native vegetation. Lawsuits emerged as farmers whose crops were damaged by drifting dicamba sued neighbors whose use caused the damage. Similar resistance patterns emerged with Bt crops. Insects evolve just like weeds. When Bt corn and cotton were first planted on millions of acres, they created enormous selection pressure, favoring any insects with genetic variations that made them less susceptible to Bt toxins. By the 2010s, resistant populations of corn rootworms, cotton bollworms, and other pests were documented in multiple regions. Scientists had predicted this would happen. Evolution is inevitable when organisms face strong selection pressure. The question was how quickly resistance would develop and whether management strategies could slow it. Regulatory agencies required refuge plantings, areas of non-Bt crops where insects could survive without selection pressure, theoretically diluting resistance genes in the population. But compliance with refuge requirements was mixed, and in some regions, resistance developed faster than models predicted. The emergence of resistant weeds and insects doesn't mean Bt and herbicide-tolerant crops failed environmentally. But it does mean they're not permanent solutions. They're tools that can be valuable, but that need to be used as part of integrated pest management strategies, not as singular solutions. The early promise that genetic engineering would solve pest problems once and for all proved naive. Concerns about GMO crops affecting biodiversity take several forms. One worry is that genes from GMO crops might flow into wild relatives through cross-pollination, creating new hybrid plants with unpredictable characteristics. This gene flow is real and has been documented. In Mexico, genes from GMO corn have been found in traditional maize varieties, despite Mexico having banned GMO corn cultivation. The genes apparently came from imported corn used for food that was planted by farmers, or from cross-pollination with nearby fields. Whether this gene flow is harmful depends on context. In some cases, it might not matter much. A wild plant acquiring herbicide resistance doesn't necessarily become more invasive unless it's actually exposed to that herbicide. But in other cases, particularly with centers of crop diversity, where traditional varieties are cultural and genetic treasures, gene flow from GMO crops represents a genuine loss. Mexico is the center of maize diversity, home to thousands of traditional varieties developed over millennia. Contamination of this diversity with genes from industrial GMO varieties concerns researchers who study crop evolution and conservationists who value agrobiodiversity. Bt crops raise interesting questions about effects on non-target organisms. The Bt proteins are supposed to be highly specific, toxic only to particular insect groups. But some studies have found that Bt toxins can affect non-target insects under certain conditions. A famous study found that monarch butterfly caterpillars fed milkweed leaves, dusted with pollen from Bt corn, had higher mortality rates than caterpillars fed non-Bt pollen. This created significant alarm. Monarchs are beloved butterflies already experiencing population declines. Subsequent research found the situation was more complicated. The original study used artificially high concentrations of pollen. In field conditions, monarchs rarely encountered lethal doses. The risk to monarch populations from Bt corn pollen appeared to be minimal compared to other threats they faced like habitat loss and pesticide use. Still, the case illustrated that Bt crops could potentially affect non-target species, and that careful monitoring was necessary. Soil ecology is another area of concern. Bt toxins don't just disappear after plants die. They can persist in soil for months or even years, bound to soil particles. Some studies have found changes in soil microbial communities in fields where Bt crops are grown repeatedly. Whether these changes matter for soil health or ecosystem functioning remains debated, with some studies finding concerning effects and others finding minimal impact. One of the most contentious environmental questions involves comparing GMO agriculture to organic agriculture. Proponents of GMOs point out that higher yields mean less land needs to be cleared for farming, potentially preserving habitat. They note that reducing insecticide use benefits ecosystems. Critics counter that industrial monocultures, whether GMO or conventional, are environmentally destructive compared to diverse, organic farming systems that build soil health, support biodiversity, and don't rely on synthetic inputs. This debate often generates more heat than light because it's comparing two different visions of agriculture. Industrial agriculture, GMO or not, prioritizes maximizing production from each acre, using technology and inputs to achieve high yields. Organic and agroecological approaches prioritize sustainability, soil health, and ecosystem integration, often accepting lower yields in exchange for environmental benefits. GMO technology fits most naturally into industrial agriculture, though theoretically it could be used in any system. The environmental impact of GMOs also depends heavily on how they're used. Bt crops grown as part of integrated pest management, with refuges planted and insecticide use minimized, have very different impacts than Bt crops grown in continuous monoculture. Roundup Ready crops used with judicious herbicide application and diverse weed management strategies differ dramatically from Roundup Ready crops drenched repeatedly with glyphosate. Climate change adds new complexity to environmental questions about GMOs. Agriculture contributes significantly to greenhouse gas emissions, and climate change is already affecting crop productivity through increased heat, changing precipitation patterns, and more frequent extreme weather. GMO crops engineered for drought tolerance, heat resistance, or improved nitrogen use efficiency could potentially help agriculture adapt to climate change while reducing its environmental footprint. Several such crops are in development or early commercialization. Drought-tolerant corn, for instance, has genes that help it maintain productivity under water stress. In regions facing increasing water scarcity, this could reduce irrigation demands. Crops with improved nitrogen use efficiency might require less fertilizer, reducing the greenhouse gas emissions associated with fertilizer production and use, as well as reducing water pollution from fertilizer runoff. Whether these next-generation GMO crops fulfill their environmental promise remains to be seen. The history of first-generation crops teaches that benefits often come with trade-offs, that evolution and ecology don't care about our technological solutions, and that how technology is deployed matters as much as the technology itself. The honest assessment is that GMO crops have delivered some environmental benefits, particularly in reducing certain insecticide uses, but have also contributed to environmental problems, particularly through heavy herbicide use and resistance evolution. They're neither the environmental disaster that critics sometimes claim nor the green solution that proponents promised. They're agricultural tools with environmental implications that depend on context, management, and integration with other practices. This nuanced reality doesn't make for compelling advocacy on either side of the debate, but it's closer to the truth than absolutist positions allow. Walk through an American supermarket and you're surrounded by GMO-derived ingredients, though you might not realize it. That box of cereal likely contains corn or soy ingredients from GMO crops. The cooking oil is probably from GMO soybeans or canola. The sugar might be from GMO sugar beets. Even foods that seem far removed from crops, yogurt, cheese, vitamins, bread, often contain ingredients derived from GMO crops at some point in their production chain. Yet if you asked shoppers which products contain GMOs, most would have little idea. This invisibility has been central to the GMO debate, raising questions about consumer knowledge, choice, and the right to know what's in our food. Let's start with what crops are actually genetically modified. Globally, the GMO crops grown at significant scale are remarkably few. Soybeans, corn, cotton, and canola represent over 99% of GMO crop acreage. These four crops, particularly soybeans and corn, appear throughout the food system primarily as ingredients rather than whole foods. Additionally, some sugar beets, papaya, summer squash, alfalfa, and a few other crops are grown as GMOs in smaller amounts. What you won't find genetically modified, despite common misconceptions, are tomatoes. The flavor saver disappeared decades ago. Wheat, rice, golden rice isn't yet commercially available in most places. Potatoes, a small amount of GMO potatoes exists, but isn't widespread. Carrots, lettuce, or most fruits and vegetables. The produce section of your grocery store is almost entirely GMO-free. When you eat whole fruits and vegetables, you're almost certainly eating conventional or organic crops, not GMOs. GMOs enter your diet primarily through processed foods and animal products. Corn and soybeans are processed into dozens of ingredients. High fructose corn syrup, cornstarch, corn oil, dextrose, maltodextrin, and dozens of other compounds come from corn. Soybean oil, soy lecithin, soy protein isolate, and various other soy derivatives appear in countless products. These ingredients are so ubiquitous in processed foods that avoiding them is difficult. About 85 to 90% of corn grown in the United States is GMO, primarily Bt corn, Roundup Ready corn, or corn with both traits stacked together. Most of this corn goes to animal feed or industrial uses like ethanol production. Some becomes high fructose corn syrup, corn oil, or other food ingredients. Similarly, over 90% of soybeans are GMO Roundup Ready varieties. Most soybeans become animal feed or soybean oil. The protein meal left after oil extraction goes primarily to livestock. This brings us to an interesting question. When you eat meat, dairy, or eggs from animals fed GMO crops, are you eating GMO food? Scientifically, no. Animals digest the feed, breaking down proteins and DNA into basic building blocks that are identical whether they came from GMO or conventional sources. Studies have found no meaningful differences in meat, milk, or eggs from animals fed GMO versus conventional feed. The genetic modification doesn't transfer through animal products. Yet some consumers want their animal products to come from animals fed non-GMO feed, and a market has emerged for such products. Fed non-GMO labels on meat, eggs, and dairy command premium prices, serving consumers who want to avoid supporting GMO agriculture, even if the products themselves are scientifically indistinguishable from conventional versions. The labeling debate has been fierce and politically charged. Proponents argue that consumers have the right to know whether their food contains GMO ingredients, regardless of safety. They point to surveys showing most Americans want GMO labeling. They note that mandatory labeling exists in the European Union and other countries without causing major problems. Transparency, they argue, builds trust and allows informed choice. Opponents of mandatory labeling, including most food industry groups and agricultural organizations, argued that labels would mislead consumers into thinking GMOs are dangerous when scientific evidence says they're safe. They worried labels would function as warning signs, tanking sales of perfectly safe products. They also cited costs of segregating GMO and non-GMO ingredients tracking them through supply chains and redesigning labels. The debate played out in state legislatures and ballot initiatives. Vermont passed mandatory GMO labeling in 2014 to take effect in 2016. Other states considered similar measures. The food industry, facing a patchwork of state laws, eventually supported federal legislation that would preempt state laws while establishing national standards. In 2016, Congress passed and President Obama signed legislation requiring disclosure of GMO ingredients, though the law gave companies flexibility in how to disclose. Through text on labels, QR codes that consumers scan with smartphones, or phone numbers to call for information. This compromise satisfied few people. Consumer advocates wanted clear text labels that couldn't be missed. Industry preferred the flexibility of QR codes that didn't flag products as somehow problematic. The resulting standard, implemented by the USDA, uses the term bioengineered rather than GMO and allows various disclosure methods. How much this actually informs consumers remains debatable. Meanwhile, voluntary non-GMO labeling exploded. The Non-GMO Project, a nonprofit organization, created a verification program that certifies products as meeting their standards for GMO avoidance. Their butterfly logo appears on thousands of products, from foods that could never contain GMOs, salt, water, to foods where GMO versions don't exist, oranges, avocados, to products where it's meaningful, corn chips, soy milk. The label became a marketing tool, allowing brands to appeal to GMO-concerned consumers regardless of whether GMO versions of their products existed. The success of non-GMO labeling reveals consumer preferences, but also raises questions about what exactly consumers are choosing. When someone pays extra for non-GMO salt, they're not getting a functionally different product. Salt is a mineral, not a living organism, and cannot be genetically modified. They're signaling values about agriculture and food systems, or responding to marketing that positions non-GMO as healthier or safer, even when science doesn't support meaningful differences. Organic certification automatically excludes GMO ingredients, as organic standards prohibit genetic engineering. This makes organic products an easy choice for consumers wanting to avoid GMOs, though organic products cost significantly more, placing them out of reach for many families. The price premium raises equity questions. If non-GMO food is important, should it be accessible only to those who can afford it? The practical reality for most people is that avoiding GMOs entirely requires significant effort and expense. It means reading labels carefully, choosing organic or verified non-GMO products, avoiding processed foods with corn or soy derivatives, and generally being hypervigilant about food sourcing. For people who believe GMOs pose health risks, this effort might seem worthwhile. For those who are reassured by scientific consensus on safety, the effort and expense might seem unnecessary. Some people choose to avoid GMOs for reasons unrelated to personal health. Environmental concerns, opposition to corporate agriculture, support for organic farming, or simply as an expression of food values. These motivations are legitimate even if health concerns aren't scientifically supported. Food choices are never purely rational or purely about individual health. They're cultural, emotional, ethical, and social. The ubiquity of GMO-derived ingredients in processed foods has also prompted some people toward whole foods and home cooking. If you're eating fresh vegetables, fruits, whole grains, and minimally processed foods, you're naturally avoiding most GMOs regardless of whether that's your intention. The Eat Food, Not Too Much, mostly plants approach to nutrition, popularized by Michael Pollan, sidesteps the GMO issue almost entirely while promoting dietary patterns most nutritionists consider healthy. What's interesting is how invisible GMOs remain to most consumers despite their prevalence. Unlike organic, grass-fed, or free-range labels that consumers actively seek out, Most people eating GMO-derived ingredients don't know it and aren't consciously choosing it. The foods simply don't taste different, don't look different, and don't cost differently than they would have before GMO crops were introduced. For better or worse, the GMO revolution in agriculture happened largely without consumers noticing or participating in the decision. This raises philosophical questions about food democracy and technological change. Should major changes in food production require consumer consent, or is it sufficient that regulatory agencies ensure safety? Do people have the right to choose non-GMO options even if the scientific rationale is weak? Should labeling be mandatory to enable choice, or does labeling mislead consumers into thinking there are meaningful differences when science says there aren't? These questions don't have simple answers because they involve values, not just facts. What we can say is that GMO ingredients are widespread in processed foods, largely invisible to consumers, scientifically indistinguishable from conventional counterparts in most cases, and increasingly available in non-GMO versions for those who want to avoid them. The market is providing options, though at different price points, allowing people to vote with their dollars for the kind of agriculture they want to support. The story of GMOs is far from over. In fact, we're entering a new chapter in agricultural genetic modification that could make the debates of the past few decades seem like a preliminary skirmish. New technologies, particularly CRISPR gene editing, are enabling modifications that are more precise, faster to develop, and harder to categorize as GMO or conventional. Meanwhile, climate change, population growth, and environmental degradation are creating urgent needs for agricultural innovation. The next generation of genetically modified organisms might look very different from the herbicide-tolerant and insect-resistant crops that dominated the first 30 years. CRISPR, which stands for Clustered Regularly Interspaced Short Palindromic Repeats, is fundamentally changing how scientists can modify organisms. Discovered in bacteria and developed into a gene editing tool in the 2010s, CRISPR allows researchers to make precise changes to DNA sequences with unprecedented accuracy and efficiency. Instead of inserting foreign genes from other species, CRISPR can edit existing genes, making changes that are often indistinguishable from natural mutations. Imagine a tomato that's naturally more resistant to drought because a single DNA letter has been changed in one gene, subtly altering how the plant responds to water stress. This change could have occurred through natural mutation. In fact, it probably has occurred somewhere in wild tomato populations. CRISPR just makes it possible to create that change intentionally in a specific variety, without waiting for random chance or breeding it in from wild relatives over many generations. This precision raises fascinating regulatory questions. If a CRISPR-edited plant is indistinguishable from what could occur naturally, is it a GMO? Different countries have answered this differently. The United States generally regulates based on the final product rather than the process, meaning many CRISPR-edited crops don't require GMO-style regulation if they could have been created through conventional breeding. The European Union regulates based on process, treating most CRISPR-edited organisms as GMOs regardless of whether the changes could occur naturally. Several CRISPR-edited crops are already being developed or commercialized. A gene-edited soybean oil with healthier fats has been approved in the United States. Mushrooms that don't brown as quickly have been developed. Rice with resistance to bacterial blight, wheat resistant to powdery mildew, and tomatoes with improved nutritional profiles are in various stages of development. These products represent a gentler form of genetic modification, tweaking existing genes rather than adding entirely new ones. Climate change is driving interest in crops modified for drought tolerance, heat resistance, and adaptability to changing conditions. Traditional breeding is working on these traits too, but genetic modification could accelerate progress. Researchers are identifying genes that help plants withstand environmental stress and working to enhance those capabilities. Some projects involve moving genes between crop relatives, while others use gene editing to optimize existing stress response pathways. The challenge with climate adaptation traits is complexity. Drought tolerance isn't controlled by a single gene, but by networks of genes interacting in sophisticated ways. A plant that survives drought does so through multiple mechanisms: deeper roots, more efficient water use, ability to close stomata to prevent water loss, osmotic adjustment at the cellular level. Engineering comprehensive drought tolerance is harder than adding insect resistance or herbicide tolerance. Nevertheless, some progress is being made. Drought-tolerant corn varieties, created through both conventional breeding and genetic modification, are showing modest improvements in water-stressed conditions. While not miracle solutions, these incremental improvements could matter significantly in regions facing water scarcity. Similarly, work on heat-tolerant crops could help maintain productivity as average temperatures rise and heat waves become more common. Nutritional enhancement represents another frontier. Golden rice was the first attempt, but numerous other projects are underway. Biofortified crops, plants modified to contain higher levels of vitamins, minerals, or beneficial compounds, could address micronutrient deficiencies affecting billions of people. Iron-enriched rice, zinc-enhanced wheat, and crops with improved protein quality are being developed. Some of these nutritional improvements use genetic engineering, while others use conventional breeding or genetic markers to select for beneficial traits. The approaches are converging, with genetic modification becoming one tool among many for crop improvement rather than a separate category requiring different treatment. Perhaps the most radical developments aren't about modifying crops, but about creating food through entirely new processes. Cellular agriculture, growing animal products without animals, represents a fundamental reimagining of food production. Companies are producing milk proteins through fermentation, growing meat from cell cultures, and creating eggs without chickens. These products involve genetic engineering of microorganisms or cell culture techniques, but the final products are molecularly identical to conventional animal products. Lab-grown meat, once science fiction, has progressed to the point where several companies have produced beef, chicken, and fish from cultured animal cells. The process involves taking a small sample of animal cells, growing them in bioreactors with nutrient media, and coaxing them to develop into muscle tissue. The result is real meat, with the same proteins and fats as conventional meat, but produced without raising and slaughtering animals. The potential benefits are substantial. Cultured meat could dramatically reduce the environmental impact of meat production, which is a major contributor to greenhouse gas emissions, deforestation, and water pollution. It could eliminate animal suffering in meat production. It could make meat production more efficient, requiring less land, water, and energy. Some advocates see it as the future of sustainable protein. Challenges remain formidable. Current production costs are high, though falling rapidly as the technology matures. Scaling from laboratory to industrial production requires solving engineering problems around bioreactor design, media formulation, and tissue structure. Consumer acceptance is uncertain. Will people be willing to eat meat grown in vats rather than on animals? Cultural and psychological barriers may be as significant as technical ones. Similar developments are happening with plant-based meat alternatives that use genetic engineering in their production. Impossible Foods, for instance, uses genetically engineered yeast to produce heme, the iron-containing molecule that gives meat its distinctive taste. This GMO-derived ingredient is combined with plant proteins to create burgers that taste remarkably similar to beef. The product is controversial among some environmental groups who oppose GMOs, even though its purpose is reducing environmental impact of meat consumption. These developments illustrate how GMO debates are evolving. The question isn't just about whether crops in fields are genetically modified, but about how genetic engineering is used throughout food systems, from fermentation organisms producing food ingredients to cell cultures producing meat, to modified plants producing improved nutrition. Synthetic biology, which involves designing and constructing new biological systems, extends these possibilities further. Researchers are working on microorganisms engineered to produce compounds that normally require expensive or environmentally damaging processes. Vanilla flavoring, palm oil alternatives, spider silk proteins, and numerous other products could potentially be produced through fermentation rather than conventional agriculture or industrial chemistry. These innovations could transform food systems, but they also raise questions about what natural means, whether food should be produced through biological manufacturing rather than farming, and who controls these powerful technologies. The debates that surrounded GMO crops will likely resurface around cellular agriculture and synthetic biology, perhaps even more intensely. Regulation will need to adapt to these new technologies. Current regulatory frameworks were designed for GMO crops that are recognizably similar to conventional crops, but with one or two new genes. How do you regulate a CRISPR-edited plant that's indistinguishable from natural variation? How do you classify meat that's biologically identical to conventional meat, but produced entirely differently? These questions are being worked out in real time, as technologies develop faster than regulatory systems can adapt. International coordination on GMO regulations has been elusive, and new technologies may make harmonization even more difficult. Some countries will embrace CRISPR-edited crops as essentially conventional, while others will treat them as GMOs requiring extensive testing. Some consumers will embrace lab-grown meat as ethical and sustainable, while others will reject it as unnatural. The global food system, already complicated by different standards and consumer preferences, may become even more fragmented. What seems certain is that genetic modification in various forms will play increasing roles in food production. Climate change, population growth, resource constraints, and environmental degradation are creating pressures that make agricultural innovation necessary, not optional. Whether that innovation should involve genetic modification remains contentious, but the practical reality is that it will be part of the toolkit, alongside conventional breeding, agroecological practices, and other approaches. The future might not involve choosing between GMO and non-GMO, organic and conventional, high-tech and traditional. Instead, different approaches will coexist, serving different markets, values, and needs. Wealthy consumers in developed countries might choose artisanal, non-GMO, organic foods, while also eating lab-grown meat for environmental reasons. Small farmers in developing countries might grow GMO crops to protect against climate-related stresses, while maintaining traditional varieties for cultural reasons. The food system might become more diverse rather than more uniform, with genetic modification as one option among many, rather than a universal solution or universal threat. What we can hope for is that future developments in agricultural biotechnology learn from the mistakes of first-generation GMOs. That development happens with greater transparency and public input. That benefits are distributed more equitably rather than concentrating in corporate hands. That environmental impacts are carefully monitored and managed. That consumer choice is respected through clear labeling. That the technology serves genuine human needs, rather than primarily generating profit. Tonight, we've traveled together through thousands of years of human food modification. What emerges from this journey isn't a simple answer about whether GMOs are good or bad, safe or dangerous, the future of food or a corporate mistake. Instead, we find complexity. We find a technology that has delivered some benefits while creating some problems. We find scientific consensus on safety existing alongside legitimate concerns about environmental impact, corporate control, and agricultural systems. We find that the same innovation can simultaneously reduce insecticide use and increase herbicide use, can feed people while concentrating power, can represent scientific progress and corporate overreach. Understanding GMOs requires understanding not just molecular biology, but also history, economics, culture, and the very human tendency to see in new technologies both our salvation and our doom. Thank you for joining me on this journey through the fascinating, contentious world of genetically modified organisms. If you're already drifting off, then our mission is complete. But if you're still awake, perhaps another deep dive through one of our previous episodes will carry you gently into slumber. Until we meet again in these quiet hours, may your food nourish both body and soul. May your choices reflect your values. And may you find peace in the knowledge that growing and eating food has always been an act of hope in an uncertain world.